Welcome to Cell Biology. Today, we are going to explore one of the most fascinating logistical miracles in nature, the nucleosome. Imagine trying to fit 2 meters of fragile thread into a microscopic box without tangling it. That is exactly what your cells do with your DNA every single second. Let us dive in and see how this incredible biological packaging system works. Now that we understand the scale of the challenge, let us look at how the cell solves it. If you just shoved 2 meters of DNA into a tiny nucleus, it would become a hopelessly tangled, knotted mess. You would never be able to find or read the specific genetic instructions you need. To solve this, you would naturally use tiny spools to carefully wind the thread around, keeping it organized, compact, and easy to manage. In your cells, these biological spools are absolutely essential. Without them, our massive genetic code could never fit inside our cells, nor could it be read properly by the cellular machinery. This brings us to the biological spool itself, the nucleosome. Why does this specific structure matter? Because it is the foundational building block of all our chromosomes. The core of this spool is made of 8 highly basic proteins, called histones, forming an octamer. The DNA, which is negatively charged, wraps tightly around this positively charged protein core. Exactly 147 base pairs of DNA wrap around it about 1.65 times in a left-handed turn. This precise wrapping is the first crucial step in shrinking the massive length of our genetic material into a manageable size. Beyond just acting as spools, these structures have another critical job. Between each nucleosome is a short stretch of linker DNA, often clamped in place by a 5th histone called H1. Under a microscope, this looks exactly like beads on a string. But these beads are not just for storage, they are the master switches for your genes. By wrapping the DNA tightly, nucleosomes hide specific genes from the cellular machinery that reads them. When a cell needs to turn a gene on, it chemically modifies the histones to loosen the DNA. If nucleosomes did not exist, every gene would be turned on all the time, leading to total biological chaos. Now that we understand what a nucleosome is, let us look at how we can measure this packaging mathematically. Biologists use this simple formula to determine the total physical length of a DNA molecule based on its nucleosomes. The variable L is the total length. N is the number of nucleosomes. The variables C and L represent the number of base pairs in the core and linker DNA respectively. Finally, D is the constant physical distance between 2 base pairs, which is 0.34 nanometers. If the number of nucleosomes increases, the total length of the DNA molecule naturally increases as well. This equation transforms a biological concept into a measurable physical reality. Before we move on, let us see this formula in action with a concrete example. Suppose we are studying a human gene that is exactly 10,000 base pairs long. We are given that the core DNA is 147 base pairs, and the linker DNA is 53 base pairs. First, we find the repeat length by adding the core and linker base pairs, giving us 200 base pairs per nucleosome unit. Next, we divide the total gene length by this repeat length, revealing that exactly 50 nucleosomes organize this gene. Finally, multiplying the total base pairs by the distance per base pair gives a physical length of 3,400 nanometers. This means a 3.4 micrometer gene is neatly compacted by 50 microscopic spools. Now that we have used this formula, let us understand where it comes from conceptually. The first chunk of our reasoning establishes the fundamental repeating pattern of the chromatin structure. We define a variable R, the nucleosome repeat length. This is simply the sum of the core DNA, C, and the linker DNA, L. Why do we add them? Because every time we move down the DNA strand, one complete spool unit must include both the wrapped portion and the string connecting it to the next one. In our example, this gave us exactly 200 base pairs. Knowing this allows us to easily divide any large, continuous stretch of DNA into discrete, manageable, repeating packages. Building on that repeating unit, we can now derive the final physical length. If we know the total number of nucleosomes, n, and we multiply it by our repeat length, we get the total number of base pairs in that entire section of DNA. But base pairs are just a count, not a physical distance. To find the actual length in space, we must multiply that total count by the physical distance between each adjacent base pair, denoted by D. This logical progression leads us directly to our final formula. It beautifully illustrates how biology uses simple geometry to solve a complex spatial problem, ensuring every single base pair is accounted for. So, we have proven how to calculate the total length of DNA. This brings us to an important question. Just how much space does the cell actually save? To answer this, biologists use a concept called the packing ratio. This ratio is a measure of efficiency. It tells us how many times shorter the DNA becomes when it is wrapped around the histone core, compared to when it is fully stretched out in a straight line. By understanding this ratio, we can truly appreciate the extreme physical compaction that prevents the cell nucleus from bursting under the volume of its own genetic material. Let us look at the mathematical form of this concept. The packing ratio, P, is defined as the length of the fully stretched DNA divided by the length of the packaged structure it forms. Here, LC represents the physical length of the core DNA if it were pulled completely straight. DN represents the physical diameter of the histone octamer spool itself. Because we are dividing a length by a length, the packing ratio is a dimensionless number. If the stretched length increases while the spool size stays the same, the packing ratio increases, meaning the DNA is packed even more tightly. Let us apply this formula to a single nucleosome core particle. We know the DNA wrapped around it is 147 base pairs long, and the physical diameter of the spool is about 11 nanometers. First, we calculate the stretched length of the core DNA by multiplying the base pairs by 0.34 nanometers, giving us roughly 50 nanometers. Next, we divide this stretched length by the 11 nanometer diameter of the spool. The result is approximately 4.54. This concrete number tells us that the genetic material is compressed to be about 4.5 times shorter than its natural length. Now that we have calculated the packing ratio, let us break down the physical intuition behind these numbers. In this first step, we're establishing the baseline state of the DNA. By multiplying the 147 base pairs by the distance per base pair, we find that the DNA wrapped around just a single nucleosome would be about 50 nanometers long if pulled perfectly straight. This is a significant length of DNA for just one microscopic spool. Without the histone proteins to organize it, this 50 nanometer stretch would be loose, highly vulnerable to chemical damage, and prone to tangling with neighboring strands. Continuing our derivation, we compare that vulnerable 50 nanometer stretch to its packaged state. The histone octamer spool has a diameter of only 11 nanometers. By dividing the stretched length by this package diameter, we see the mathematical proof of the cell's efficiency. Just by wrapping the DNA around the histone octamer, the cell compresses the genetic material to be about 4.5 times shorter. This is the crucial first step in DNA compaction. It is a brilliant evolutionary solution that ultimately allows meters of DNA to fit inside a nucleus that is only micrometers wide. Let us bring all these concepts and numbers together visually. On screen, you are looking at a detailed schematic of a single nucleosome. In the center is the histone octamer, the protein spool made of H2A, H2B, H3, and H4. Notice the double helix DNA wrapping tightly around it. This is the 147 base pairs of core DNA we calculated earlier. Below the spool, you can see the physical diameter labeled as 11 nanometers, which gives us our packing ratio. On the right, the DNA extends outward as linker DNA, connecting to the next spool. Finally, sitting right at the edge where the DNA exits the core is histone H1, locking the structure in place. This diagram perfectly captures the fundamental unit of chromosome structure. We have covered a lot of ground today, so let us review the most important ideas. First, the nucleosome is the foundational building block of chromosome structure, acting as a microscopic spool made of 8 histone proteins. Second, it solves a massive logistical problem, allowing 2 meters of DNA to fit into a tiny nucleus while keeping it protected and organized. Third, by controlling how tightly DNA is wrapped, nucleosomes serve as the master switches for gene expression. Finally, as our calculations showed, this biological packaging system is incredibly efficient. Understanding the nucleosome is the absolute key to understanding how your genetic code is stored and utilized every single second of your life. Great job following along.